Hello and welcome. Today, we're exploring a surprising connection: how the mathematical field of knot theory helps us understand the tiny, tangled world of molecules. Yes, the same kind of knots you might find in a shoelace, but on a microscopic scale, influencing DNA, proteins, and polymers. Let's start with DNA. Inside our cells, DNA strands are incredibly long and can become knotted and tangled. These knots aren't just messy accidents, they're important for how DNA works. Enzymes called topoisomerases act like molecular scissors and glue, managing these tangles. Knot theory allows scientists to classify these DNA knots, understand precisely how these enzymes work by seeing how knots change, and study how DNA is packed so efficiently into tiny spaces like cell nuclei. It even sheds light on how knots form and resolve during essential processes like DNA replication and recombination. For instance, the way daughter DNA molecules are linked after copying tells us about how that process finishes. Now, what about proteins? These are the workhorses of our cells, with complex three-dimensional shapes. While true knots in a protein's main chain are rarer, they do exist and can be vital for the protein's stability or job. The presence of a knot can contribute to a protein's stability, folding pathway, or function. Knot theory helps identify these knotted proteins and understand their role. It also helps us analyze how proteins fold into their specific shapes, navigating through tangled states, and how their structure impacts their stability and dynamic behavior. Beyond biology, knot theory is a game changer in polymer physics. Think of long-chain polymers, whether natural, like cellulose, or synthetic, like plastics. In solutions or when melted, these chains inevitably get tangled. Knot theory helps quantify this entanglement, which is key to understanding properties like how syrupy or stretchy a material is. It aids in modeling how polymers move and respond to forces. The likelihood of knots forming in polymers can even signal phase changes, like when a polymer coil collapses into a globule, affecting material properties. And recent research hints that dense systems of unknotted ring polymers might get stuck, forming a topological glass, simply because the rings can't pass through each other. Looking ahead, the application of knot theory in molecular biology and polymer physics is rapidly growing. With better computers, advanced experimental techniques like single-molecule manipulation, and new theoretical tools, we're pushing the boundaries. Future research will likely delve deeper into the link between a molecule's topology and its function in more complex biological settings. We might even design new biomaterials with specific topological features and further uncover how molecular machines manage these intricate structures. The development of new mathematical tools will also continue to provide deeper insights into the fascinating world of molecular knots and links. Thanks for tuning in.